Welcome back to Tim D's USMLE Step Prep Biochemistry Lesson 2, DNA replication and how DNA repairs itself. So, let's get right started. When talking about DNA replication, that just means DNA is making DNA with DNA. So it's making more of itself. It's replicating itself. It's duplicating itself. How does it do that? Well, you need to split it apart, and all these enzymes and proteins are going to help make that copied strand. And you should have a direct copy of the DNA afterwards. So you'll start out with one template strand of DNA and end up with two daughter DNA strands. So, first thing to keep in mind is that DNA replication is way different in eukaryotes, in humans or animal cells, than it is with prokaryotes, which are the bacterial organisms. Now, we have to understand that bacteria, to start off, have circular stranded DNA. Therefore, they only have one site of the origin of replication. Whereas when we have human DNA, there's multiple origins of replication in human DNA that occur along the strand. So where does DNA replication happen? Well, it happens in the nucleus of our cells. And when does it happen? Well, if you guys remember that cell cycle that we saw in lesson 1, we saw that during S phase is the synthesis phase, and DNA replication occurs during the synthesis phase because you got to synthesize more DNA in the synthesis phase. DNA replication is bidirectional. So when it occurs, replication is happening on both sides. It's as if a zipper is opening and being opened on both sides. And one of the important zippers, the opener, is helicase. We'll talk a little bit more about helicase in a minute. But now I just want to hit you guys with a couple names of some of these enzymes that are going to come in handy when we go about talking about what their job is, what they're actually doing that involves replication. So two of the main things that we're going to talk about today are polymerases and nucleases. Now, polymerase, those are what's going to synthesize the nucleic acids on the strand by forming phosphodiester bonds. And then the nucleases, on the other hand, are what's going to snip off the nucleic acids and by snipping their phosphodiester bonds. These are going to be very important, and let's take a look now at how these work. So let's break down replication step by step. All right. So the very first step that happens is that the origin of replication is going to be recognized. Those base pairs are going to signal that this is where the origin of replication is. Now, the next thing that's going to happen is the zipper is going to come along, helicase. Helicase is going to break the hydrogen bonds that holds the base pairs together, and this is when the two parental strands of DNA will begin to unwind and form the two replication forks, right? So DNA is stranded like this. Here's the origin of replication. Helicase is going to come in and slowly start to create two forks of replication that are going to start spreading out. So that's what gets it started, right? And then we have a very important protein come along called single-stranded DNA protein, SSB. is what it's called for short. And this guy is just there to make sure everything goes all right. So he's going to connect to that strand when it's just open and sitting there, to make sure that it's not degraded, to make sure that it doesn't break off, to hold it in place. Basically, he's going to prevent them from reassociating them and protect them from being degraded by nucleases. So he's important, the single-stranded binding protein. So the next thing that has to happen is that an RNA primer has to be laid down first. It really does. The RNA primer is this small band of nucleotides of RNA that is basically it's synthesized by primase, and primase makes the primer. And the primer is huge. We need the primer in order to start synthesizing the DNA. And I'll get to why that is in a second. But so the primase is going to make the primer, and the primer is going to go and attach and lay itself down on the template strand. Template strand, parent strand, same thing. It's the template, right? It's what you're copying. When it lays down the RNA primer, it's going to lay it down in the 5' to 3' fashion. And it begins at the origin of each strand. So why is it important to put the primer down? Put RNA primer on the parental strand, on the template strand. It's super important because DNA polymerase 3 to be exact, needs to see this primer because it's going to synthesize off the 3' end of the primer. So it's very important that the primer is there because DNA synthesis just can't start without a primer. It's got to know where to go, and it's got to know what to start synthesizing. And thank God for primase for making those primers so that, we can see that happen. So DNA polymerase 3 will start synthesizing the DNA, grabbing nucleotides, putting them right there, in a 5' to 3' direction, starting off the 3' end of the primer. And if that makes sense, I hope it does. I'll show you guys this picture now, so that you can see the 5' end and the 3' end, and DNA polymerase is working this way to add on those nucleotides to synthesize the DNA. Now obviously all double-stranded DNA is anti-parallel, so as you guys can see, it's going to make off of the template strand, it's going to make the new synthesized strand an exact anti-parallel copy. So in order to put these nucleotides up on the strand, DNA polymerase is going to need high energy nucleotides. So it doesn't want no monophosphate nucleotides. It doesn't want no diphosphate nucleotides. It needs those triphosphate nucleotides, the high energy triple phosphate molecules. So it's looking for the deoxyguanine triphosphate, deoxyadenine triphosphate, deoxycytosine triphosphate, and deoxyadenine triphosphate, I think is the last one. Thymine triphosphate. I might have said one of those too much. Okay, so you got DATP, DGTP, DCTP, DTTP. There you go. Those are the 4 that are obviously needed for DNA. You guys learned that in the first lesson. Now, the strand that's made completely in one long piece is called the leading strand. It's not really important for the USMLE to know, to be honest. It's simple biochemistry. But that's what's called the leading strand. There's also a lagging strand. So the lagging strand, on the other hand, is not continuous. It's actually discontinuous. It only does little short sections, but still about 1,000 nucleotides long, that are called Okazaki fragments. Now, just like DNA polymerase 3 was making the synthesized DNA on the leading strand. On the lagging strand, it still goes in the same direction, just off of the Okazaki fragments. So it's still going from the 5' end to the 3' end, synthesizing DNA. So for each of the replication forks that are formed by helicase, there starts off the leading strand and the lagging strand for each origins of replication. So RNA primers, once the job's done, is removed by RNA ACE H. It's specific to eukaryotes, and some DNA polymerase, it's unspecified which one actually, will come and finish the job and will synthesize the RNA within the gaps. So once the DNA polymerases are done, an enzyme called ligase will fill those small little cracks that the DNA polymerases may have missed. DNA ligase is specific to the little spaces on the lagging strand because the lagging strand has those Okazaki fragments that are only 1,000 nucleotides long. So those little gaps in between the Okazaki fragments are filled in with the help of ligase. So then there's this enzyme called topoisomerase 2. The enzyme topoisomerase 2 is what will help prevent supercoils. All you got to remember about topoisomerase 2 is that it relaxes the DNA. It's going to remove those supercoils, make everything nice and even and good to go. And topoisomerase 2 is going to go ahead of the program, ahead of the job that's being done, and ahead of the helicase that's unwinding everything. And it's going to make sure that the DNA is not supercoiled and it's not too tightly bound or loosely bound. It's got to make sure that it's good to go. So topoisomerase 2 is going to relax the DNA, make it even, make it nice, make it good to be. You know those zippers where you can't open them because it's just something's not right? Well, topoisomerase 2 prevents that from happening so that everything comes apart nice and smooth. In prokaryotic cells, topoisomerase 2 is also referred to as DNA gyrase. And it's called that because it's a completely different structure than topoisomerase 2, but they're still referred to as topoisomerase 2 in both prokaryotes and eukaryotes. Just call it DNA gyrase if you're talking about prokaryotes, it'll make it easier. So that was just the basic step-by-step. Let's get deeper and let's really understand this now, okay? So what did we just kind of mention? So the very first thing that happens once the DNA gets opened up is primase is going to make the RNA primer. And the RNA primer is going to go and attach, starting from the origin of replication, and it's going to lay it down in the 5' to 3' direction, beginning at the origin of each parental strand. So as you guys see this parental strand here, you can notice that the letters on it are the nucleotides, right? Those are the nucleotides in a sequence on the template/parental strand. An RNA primer is going to be laid down on top of it. So next, DNA polymerase is attached then to the RNA primer, and it's going to start synthesizing the nucleotides to add on. So all DNA polymerases read the DNA from 3' to 5'. The cool thing about DNA polymerase, as opposed to RNA polymerase that I'll talk about in a second, is that DNA polymerase has a spell check, a proofreader built into it. And if it didn't, we'd be pretty mutated all the time, because the DNA that's being made has to be made sure that these nucleotides are lining up. Because if the nucleotides don't line up right, what happens? If you have a guanine pairing up with a thymine, what might happen? It's not the right pair. Guanine pairs with cytosine, right? So they're not going to form that hydrogen bond. They're not going to form that strong bond. DNA is going to become unstable. And when DNA is unstable, it really can't be made into RNA, and it really can't be made into proteins. And if it does, it's going to make a faulty protein. It's going to make a faulty RNA, which is going to then make a faulty protein. So you need to have that crispy, clean, good DNA so that no pathology is going to arise, right? And that's why DNA polymerase, the type of synthesis that it makes for DNA, is called high fidelity, because there's very little mistakes. It's not perfect fidelity. There are going to be some mistakes, but it's very high fidelity. Now, during DNA synthesis, what's actually going to proofread what the DNA, while the DNA polymerase is making things? What's going to make sure that everything's right? Well, that's when the nucleases come in handy that we mentioned earlier. Now, there's two different types of nucleases. There's endonucleases that cut within, and the exonucleases that are used to pull out something, right? To pull out the nucleotide that's mismatched. It's not the right pair, so it's going to say, hey, this thymine is not supposed to connect to this guanine. Let me pull it out. So that's what it does. And that specific exonuclease is called 3' exonuclease. It's kind of written like 3' arrow 5' exonuclease, but everybody just calls it 3' exonuclease. That's the proofreader. Now, what's some of the pathology that happens if there is no exonuclease to pull that mismatched thing out of there? What's one of the pathologies you might see? Well, one of the more important ones that you'll always see is sickle cell anemia. Another one you may see, cystic fibrosis. So I was just talking about DNA polymerase the whole time. But did you know there's also polymerases for RNA called RNA polymerases? These are basically the same as DNA polymerases with a few minor differences. Now, for one, is that they both read the template strand from the 3' to the 5' end.
That's really unique, Faye.
Now, they're going to add to it, obviously, it'd be the anti-parallel way. So then that they would add to it, they would synthesize DNA in the 5' to 3' end. So every time a nucleotide is going to add itself to a strand, whether it be the template strand on DNA or RNA that's being synthesized, that high energy triphosphate nucleotide is going to throw off a couple phosphates, PPI, inorganic pyrophosphate is what's that called, and that's going to be released. So energy is released off of making DNA. Now, the nucleotides that DNA polymerase needs are a little different than nucleotides that RNA polymerase needs. So the substrates, as they're called, for DNA polymerase are going to obviously be for DNA, so they're deoxyribonucleic acid synthesizers. So they're deoxynucleotides. That's why the little D is in front of the name, like I showed you guys. No, but for RNA polymerase, doesn't need the deoxygenated ones. It'd rather have the oxygen group on there, right? Because it's RNA. So yeah, they don't have the little D on there. It's just going to be ATP, ATP, UTP. So a huge difference between RNA polymerase and DNA polymerase is that RNA polymerase does not need a primer. It doesn't need a primer to get going. And another huge difference is that it does not have a proofreader. RNA polymerase is just throwing nucleotides up on the strand. It doesn't really care about them too much. And is that a huge problem? It's a problem, but it's not really a huge problem because RNA has a very short half-life and DNA makes the RNA. So if you've got good DNA, you may have made a bad car, but the model for that car is on point. And this one car may have one accident down the line, but this car is going to be remade anyways, so don't worry about it too much. So that's the nice thing to consider, at least, that it doesn't have a proofreader. So you're going to really get, it won't really show up as a lot of pathology. And the fact that it doesn't have a proofreader and it sometimes makes errors in matching of the nucleotides, that's why it's called low fidelity RNA synthesis. That's DNA replication, everybody. It's not that bad. You got the primase making the primer, and the DNA polymerase is gonna attach to the primer. Now it's gonna need some energy. It's gonna need those triphosphate nucleotides. It's gonna add them on. It's gonna use exonuclease to make sure the job was done right. And it's gonna remove anything that isn't. And topoisomerase 2 is making sure those strands are nice and smooth. Now, what happens once DNA replication is completed? Because once it's completed, there's this little problem. And this little problem is called the end replication problem. Because at the end of replication, there's a problem. Now the problem is that at the end of the chromosome is a certain region. And this region is called the telomere. You guys see the chromosome, little X guys, the little tips of them is the telomere region. Now, why is this a problem, that there's a problem happening in the telomere region? Because every time DNA polymerase is done replicating, it actually stops short. It doesn't finish the complete strand. And that's a problem, because if there was nothing to make up for that last little strand, after every cell division, after every replication, that there's gonna be a shorter and shorter and shorter DNA. And eventually we would just cease to exist. So that's the end of replication program. Now what's gonna stop that from happening? There's one enzyme called telomerase that's going to finish the job of the DNA polymerase. And it's very, very important, obviously, but it does this through reverse transcriptase activity. Now, this shortening that occurs still happens a little bit, and that's what causes aging. So as the chromosomes become a little bit smaller in the telomerase region, it's normal to happen, because that's just what happens with age. And there's a certain very rare disorder to where this activity of telomerase is very, very little. So these things are allowed to get shorter and shorter and shorter because the telomerase isn't there to synthesize the telomeres. Now, what genetic disease am I talking about? That's pretty rare, but they're definitely going to ask about. It's called progeria. And in progeria, you have a decreased amount of telomerase activity. On the other hand, what very common disease that's not caused by this, but it's associated with the disease, occurs all over with increased telomerase activity. Cancer. Now, in cancer, the activity of telomerase is through the roof. It's creating too long of chromosomes, and that's why there's way too much cells being made and DNA and RNA being replicated, and there's just all these things going on. It's too much. Too much telomerase activity causes cancer. Too little telomerase activity causes progeria, or premature aging. The increased length of telomeres is what contributes to the immortality of malignant cancer cells. Telomerase is really only active in germ cells, so the reproductive cells, stem cells, as well as embryonic cells, but not in somatic cells. So one of the cool things about telomerase is that it does all these things through reverse transcriptase activity. So this is referred to as HTRT, telomerase reverse transcriptase activity. Speaking of reverse transcriptase activity, there's an actual DNA polymerase that's called RNA transcriptase. And it's a little similar to the other DNA polymerases that it synthesizes DNA, but it requires an RNA template. Transcriptase is used by retroviruses, such as HIV, to use to replicate their RNA genome. So the fact that this uses reverse transcriptase activity, much like telomerase also does, that means it could be subject to anti-HIV, anti-reverse transcriptase drugs, such as AZT, DDC, and DDI. Now, just to talk about pharm for one sec so you understand the biochemistry behind this, AZT works by putting a azide group, which is a triple nitrogen group, into the nucleotide to stop the whole synthesis of DNA in the first place, because it's going to be like, whoa, what is this? I'm not going to continue synthesis. This is messed up. It's not normal, right? So the replacement of the azide instead of the normal hydroxyl group at the 3' position of the deoxyribose prevents the further replication, and that's what's going to terminate the chain, cause chain termination. Now, although it is a DNA polymerase, reverse transcriptase lacks proofreading. It doesn't have the 5' exonuclease to come and help it out. So it's a good thing though, because it stops the synthesis right there, and that's how we can combat these viruses. So when we're talking about pharmacology now, there were certain drugs that were developed to block some of these enzymes' activity. Now, specifically, they were made to block bacterial's activity with the same enzymes that they use, right? So like there were the quinolones that were made a long time ago that were meant to block and stop the DNA gyrase, topoisomerase 2 in prokaryotes, DNA gyrase, to stop its activity so that it would not relax the DNA, the supercoils would not be smoothened out, and crappy RNA and crappy proteins, faulty proteins would be made because of that. And that would stop the bacteria from being able to do anything really. It would prevent their DNA transcription and translation. And most importantly, it would prevent their DNA replication. A little down the line, a new type of quinolones was made called fluoroquinolones. And these fluoroquinolones are a little bit better at stopping the DNA gyrase for prokaryotic topoisomerase 2 from synthesizing DNA. And you guys have probably heard of these. So levofloxacin, ciprofloxacin, moxifloxacin, all the floxacins. Now, do these have an effect on normal eukaryotic topoisomerase 2? Do they have an effect on our synthesis of normal DNA because we're getting rid of them supercoils? The answer is no. And why? Because I already told you guys. Topoisomerase 2 is structurally different than topoisomerase 2 in prokaryotes, the DNA gyrase. It's a totally structurally different thing. So that's why we're okay to use these drugs on each other. The fluoroquinolones will not stop our DNA replication. But speaking of stopping our DNA replication, what type of drugs are used to do that? Anti-cancer drugs, anti-neoplastic drugs. So there are certain drugs that target our topoisomerase 2, the eukaryotic topoisomerase 2, and prevent it from smoothing out and relaxing our DNA. And what drugs would those be? I'll give you guys two of them, etoposide and teniposide. Now, these are going to block the eukaryotic topoisomerase 2. They're going to not allow the DNA to become smoothened out. So, and supercoils are going to be formed. DNA is going to be unstable. RNA is not going to be made right. Proteins are definitely not going to be made right. So, do you think that's going to really affect us a lot? Do you think it's going to cause a lot of pathology? Yes and no. So the good thing is that topoisomerase 2 is only being active in cells that are in which phase of the cell cycle? Now, I hope you said G1 phase. Now, most of the cells in the body, like I explained last lesson, are in G0 phase. They're in the resting state. So their topoisomerase 2 in the nucleus is shut down. Like it's there, but it's not active. But in G1 phase cells, topoisomerase 2 is active. So if you take drugs that are going to block that, the G1 phase cells are going to be affected too. So how is that going to manifest itself now as a pathology? Well, that's where a lot of these side effects come from. So you remember you have cells that are always in that are in G1 phase most of the time are your hair follicle cells. So now when you take cancer drugs, you understand that you're going to lose your hair, right? Because the DNA is being messed up there because it's the topoisomerase 2 is being inhibited. Its activity cannot do what it's supposed to do, so now you're losing your hair. Or what's another thing that you experience when you're taking cancer drugs? A lot of GI problems. Those the cells in your GI system are not having their DNA supercoils removed, and their DNA is becoming unstable now because you're taking these anti-neoplastic drugs. So it's very important now to realize that, wow, all these side effects are coming because these drugs are going to affect certain cells in certain different phases of the cell cycle. Another thing I forgot to mention real quick is just that you have a lot of bleeding problems when you have anti-neoplastic drugs in your system, because what other cells are in G1 phase that are affected? Red nucleated cells. That's where the bleeding problems come in. Whew! That was DNA's replication. You guys just completed the DNA replication portion, and I'll be back with DNA repair. Thank you guys. Let's take a little break. So, when talking about DNA repair, a couple things you need to consider. How does DNA get damaged? How does DNA repair itself or get repaired? So, how does it get damaged? What are some things that will damage your DNA? Well, in the first lecture, we talked about heat and how heat will denature our DNA. That's why you boil stuff to clean it off, right? Because it's gonna fry up those bacterial DNA filled organisms, and the heat will denature them. But mainly the two things that'll really damage your DNA to where it needs to be repaired is chemicals and radiation. Those are the top two things. So let's get right into a pathology to give you guys an example. Let's say I'm out in the sun all day and I'm just, I don't produce a certain enzyme that is going to repair my DNA from UV radiation. What am I gonna get? I'm gonna get blisters, and my skin is just gonna be all damaged and broken and bleeding, and it's very sad to see individuals have to go through this type of thing where they have to hide from the sun all the time, and that's called xeroderma pigmentosum, and it's a pretty rare condition, but they're lacking a very important enzyme. Do you guys know which enzyme that is? The enzyme is called excision endonuclease. Now, excision endonuclease does what? It removes the thymine dimers that are formed when your DNA gets exposed to UV radiation. What the heck are thymine dimers? This is what they are, right? So you have thymine strands in your DNA, and if two thymines are next to each other and those cells are in G1 phase, they're getting ready to synthesize, so they're just sitting around waiting to get synthesized. And cells in G1 phase are very vulnerable to radiation. So those cells are not going to be able to, if they don't have excision endonuclease, stop those thymines from connecting. And those thymines form a dimer, which is going to produce unstable DNA, right? So your epithelial cells in your skin cannot stop themselves from having unstable DNA. So it's not going to be a stable composition of skin, so it's going to even have more damage from the UV radiation that's coming in there. Now, the repair enzymes in this case would be DNA polymerase and DNA ligase. We talked about this. Those are what's going to help repair that damaged part of the DNA once it's been excised from excision endonuclease, that is. So there is some repair going on, even though these thymine dimers can occur in someone that doesn't have xeroderma pigmentosum, and the excision endonuclease will do its job, take out those thymine dimers, and then DNA polymerase and DNA ligase is going to come in there and patch it up. So what happens if your DNA gets mismatched and it's supposed to be repaired normally, but it's not being repaired? Well, that's probably due to the fact that you're not producing a specific enzyme because you have a mutated gene. Now, what mutated gene would not create these enzymes? If you have a mutation on one of these two genes, then you're not going to be able to repair the mismatched bases. Now, the two genes are HMLH1 and HMSH2. And this, unfortunately, is going to result in a very unfortunate type of predisposition to cancer. And it's called hereditary non-polyposis colorectal cancer, HNPCC, otherwise known as Lynch syndrome. Now, those with Lynch syndrome have a very much more likely chance of developing colon cancer, breast cancer, even brain cancer. It's just bad. Lynch syndrome is not good to have because you can't repair these mismatched bases. The thing about Lynch syndrome is it's going to be asymptomatic. There's not going to be really any symptoms that are going to show up until you have cancer. So there have been studies done now that are pretty cool in the fact that they're thinking that they can now start screening for Lynch syndrome. So I encourage you guys to research that, because it's still kind of underway. But doctors are becoming more and more confident in being able to detect early on if somebody may have Lynch syndrome or not. So these mismatched bases are occurring after synthesis, right? Because they're mismatched. So these cells are going to be in G2 phase. So now you can kind of see how after G2 phase comes mitosis, these cells are going to divide, and some of them now may be cancerous, and the cycle then continues. So another type of damage occurs through heat. And what happens specifically is that the molecule cytosine becomes deaminated. So it loses its amine group. It's called cytosine deamination. And when cytosine becomes deaminated, what does it become? Well, it's got that amine group. The amine group comes off. Now it's just uracil. So the fact that you have uracil now, which remember is only an RNA, is going to cause a lot of structural and functional problems. So heat can cause this, and you know what also causes it? Nitrates. Nitrates are in a lot of different meats. They're in hot dogs, all kinds of nasty stuff. So heat and nitrates can cause cytosine deamination. Nitrates are also in a lot of meat preservatives. Gross. Now, there is an enzyme that's lacking in people that are really damaged by this because they can't remove the uracil that's formed through cytosine deamination. And it's called uracil glycosylase. And AP endonuclease also removes that uracil group. Now, when bases are mismatched, or when thymine dimers are formed, these DNA strands cannot form hydrogen bonds with each other. So the DNA is going to be unstable, and that's where this whole damage comes into it. Your DNA has to be stable, it has to be even, it has to be symmetrical. So now, guys, I want to talk about some very specific tumor suppressor genes that are automatically within us to make certain enzymes or proteins that are going to stop a tumor from happening. Tumor suppressor genes. They're going to stop a cancerous cell from continuing doing its thing. It's going to prevent cancer in the first place, right? And the first one to consider is P53. The P53 gene that makes P53, which P53 is known as the guardian of the genome. P53 is badass because it's sitting there right at the cell cycle when cells are about to enter into S phase. It goes, I gotta make sure you're not cancerous, or else you're not getting into the synthesis phase. These G1 cells are coming up to P53 to make sure they got the right composition before they can even continue. So P53, if they aren't meeting the requirements to get into the S phase club, then P53 is going to tell them to kill themselves. And they're going to have apoptosis. Apoptosis is programmed cell death. So cancer is when cells just will go right in to S phase, regardless of a hormone signaling, and sometimes regardless of P53 being present or not present. But if P53 is not present or it's deleted, then you could have a syndrome. Now, what syndrome am I talking about? Lee-Fraumeni syndrome. And a lot of small, hard tumors can form because of this. ATM gene is the next very important tumor suppressor gene. And it encodes a kinase that's essential for p53 to be active, to have its activity. Now there's a specific condition called ataxia telangiectasia, where ATM is inactivated. And this is when you're going to have patients that are very hypersensitive to x-rays, and they're going to have a predisposition to lymphomas. To name a couple more tumor suppressor genes real quick, we have BRCA1 and BRCA2, and without these around, the patient is very likely to develop breast cancer. Without BRCA1 specifically, they can develop breast cancer, prostate cancer, ovarian cancer. BRCA2 is primarily just breast cancer. Now, the last tumor suppressor gene I want to talk about is called RB, retinoblastoma gene. And this is basically the reverse cell cycle gene. And this is basically going to be the negative regulator of the cell cycle through its ability to bind to transcription factor. And the transcription factor that it binds to is E2F. And it represses the transcription of genes that are required for S phase. Okay, guys, thank you again for joining me today. It was a long lecture, but we got through it. You guys now know about some very common problems that occur if your DNA is not repaired. And you guys definitely know that DNA replication. So thank you. Check out the material I provided on here and tune in again and have a good night. Thank you guys. Bye.